Hey guys, welcome back. Today I am here with another topic of developmental biology, which is cell specification, types of specification and determination. So we'll discuss it one by one. So let's start. So the first is cell specification. It is also known as cell differentiation. So what it means is, suppose we have a group of cells which is stem cells. As we know that stem cells are undifferentiated cell which leads to form various cells within the body, right, like nerve cell, muscle cell, blood cell, right. So in specification, specific cells meant to perform certain tasks within the body, means a particular cell need to perform a particular function within the body, right. So first stage is a labile stage. As we know, labile mean to change. So when a cell need to perform its function, it need to change its current stage. So that stage is known as labile stage. Right, so the fate of a cell or a tissue is said to be specified when it is capable of differentiating autonomously when placed in a neutral environment such as petri dish or test tube. Means when cells is capable to differentiate by itself. It does not require its surrounding cells to perform, or any signals or external environment to perform, when it is determined to perform its function, right. It When it does not require anything to perform, when it is capable of differentiate autonomously or by itself, right. So in specification, cells are to perform reverse or change its fate, right. So it can only change its fate when it is not specified, otherwise it can change its fate anytime, right. So there are three types of specification. First is autonomous specification, second is conditional specification, and third is Syncytial specification. Right. So we'll discuss it one by one. So the first is autonomous specification. It produces a determinative or mosaic development, means the cell in this specification is already determined to perform its function, right. It does not need external condition or external cells or external signal to perform its function, right. So if a particular blastoma removed from the embryo early in its development, that blastoma will produce the same cells that it would have if it was still the part of embryo when placed in the other embryo, right. That is, cell cannot change its fate if particular blastoma is lost, right. So suppose we have this is first embryo and this is the second embryo, this is host embryo, where we need to place a particular blastoma, right. We take particular blastoma from the first embryo and we placed it by the host embryo here, right. So so the particular blastoma does not change its function or its fate where it should, but it should be in further, right. So it does not change its function, right. Whether we placed it in another embryo, right, it does not affect by surrounding cells or surrounding conditions, right. And the cell where it cell in the first embryo where it is lost, right, the cell surrounding here by the loss side, it does not compensate its loss, right. It does not compensate its loss, right. So the cell which are determined or which are capable to perform by itself, right, these are known as autonomous specification, right. So in this specification there will be no compensation by surrounding cell in the loss side, right. The loss is not compensated by surrounding cell, right. So next is, this is the diagram of a autonomous specification, right. This is the isolated trochophore development, right here. So the cell is what it should be here, right. So this is autonomous specification. Next is conditional specification, right. It is also known as regulative specification. So in this case, it allows the cell to change its fate to acquire different function, right. It means it is not determinated. It can change its fate anytime by itself, right. It is affected by the surrounding cells or surrounding condition, right, where it should place, right. So if a particular blastoma is removed from an early embryo, the remaining embryo cell alter its fate so that the role of missing cell compensate or take over, right? Suppose we have a embryo here and we have a host embryo. And when we place a particular blastoma from the first embryo to the second embryo, right here, from here, so these cells taken from here can change its fate and and acquire the condition which this embryo cell made, right? It can change its fate as it by surrounding cell, what the surrounding cell make, or what right. So it is affected by the surrounding cell and can take over the function which is acquired by the surrounding cell, right? So and the lost in the first embryo can compensate by the surrounding cell, right? So it is not determined determinative, right? It can change its fate anytime by itself, right? So this is the diagram of conditional specification, right? So this is the blastula. So these are the normal back cell, which is going to make the back tissue, right? These cells are taken from here and transplant into the normal back cell to the valley region, right here, and the cell from the valley tissue, right? These cell can change its fate and make the valley tissue here, right? So it means the in conditional specification it can change its fate anytime, right? So glass needle in this glass needle removes, right, some cells, right? So it does not affect it all, right? These lost cell can compensate by itself and the normal development of this embryo would take place, right? And the third one is known as syncytial specification, right? So it mostly occur in insects. In this case, cell fate are decided by condition affecting nuclei in a single multinucleated cell. So in this case, what happened here, the cell fates are decided by the nuclei present in the cytoplasm, right? So a cytoplasm that contain many nuclei is known as syncytium, right? So in Drosophila, the anterior most portion of egg contain an mRNA that encode a protein known as vicoid. The anterior most portion of the egg Drosophila in Drosophila is known as vicoid, which make head region, right? And the posterior most is known as hunchback, which make tail region. So in this diagram, it shows the syncytial specification. Here is the mature egg of the Drosophila, right? So the anterior most portion show bicoid mRNA and the posterior most show nanos mRNA, right? So it is the anterior most here is defines the anterior most gradient of vicoid protein and the posterior most gradient of nanos protein. So vicoid function is to make the anterior region, right, like acron means head, acron, head and thorax, right? And the nanos function is to perform abdomen, right? Is to make abdomen region, talson and the pole cells, right? So next is and the last is the determination in cell specification. So as we already know that determination means when the cell is determined to perform its function, right? It does not require any external condition or external cells or any signal to perform, right? So a cell or a tissue is said to be determined if it is capable to differentiating autonomously even when placed in the another region, right? So it is same as the autonomous specification as it does not require any external environment to perform, right? So this is all about the cell specification types and determination, right? So if you please like the video, do subscribe, share and like. Thank you.